Bismillahirrahmanirrahim. Assalamu alaikum. It's chapter number 16, Inherited Change, which is the for class A2. Now, what is the mutation? It is a change in the DNA that the cause is the mistake in replication or damage to the genetic message. Actually, mutations in somatic cells do not pass on to the offspring, so have only little evolutionary consequences. Mutation in the germline cells or the sex cells is passed to subsequent generation, thus providing the raw material from which the natural selection produces the evolutionary changes. The types of mutations, there are two types. Number one is the chromosomal mutations or chromosomal aberrations. These are actually the mega changings. This includes the loss of chromosome or addition of chromosomes. And just like deletion, insertions, inversions in the part of the chromosome. For example, in case of the Down syndrome, which occurs both in male and female, there is a condition trisomy at 21st chromosome, mean 3 chromosomes are present instead of the 2. And the Turner syndrome, which occurs in female, in which one X is missing, so only a single X is present. Klinefelter syndrome occurs in male, in which there is an extra X by providing XXY and 2 extra X instead of XY. As you know that in the male there is the XY chromosomes. There are XY chromosomes. So in the Jacobs syndrome that occurs in male, instead of XY, it has extra Y chromosome. The meta female, they have extra X chromosome, and along with XX, they have extra X and Y. So these represents the chromosomal aberration, and these are the mega changings. You can see over there that there might be the loss of chromosomes or the addition of chromosome in these cases. So there could be the change in the number of the autosome, just like in the Down syndrome, and the change in the number of the sex chromosome, like the other ones. Now, why do these changes occur? Actually, in meiosis, there is in meiosis there is actually in the anaphase and telophase there is the chromosome fail to segregate both in meiosis 1 and meiosis 2. How? For example, there are two chromosomes. In meiosis 1 there is the reduction division. What happens due to the you can say the chromosome failed to segregate? This chromosome should move into this cell and this should move into this cell, but it doesn't happen. Both move into the same cell, so there is no reduction division over this side, but this cell will remain empty. So what will happen in the next case? There should be one chromosome in each cell. The meiosis 1 is actually the reduction division, whereas the meiosis 2 is representing the same kind of division as present in the mitosis. So actually what will happen that these cells will remain, will receive no chromosome. So if there are the gametes, what will happen? This will receive extra one chromosome. This will receive extra one chromosome. This will miss one specific chromosome. This will miss one specific chromosome. So in this gamete, 23 plus 1, 23 plus 1, and 23 minus 1, 23 minus 1. Whereas, for example, in this case, for example, there are in meiosis 2, there are two chromosomes, both actually there is a process of reduction division in meiosis 1. So it will receive the half amount of chromosome as usual. But what will happen next? When there is the process of meiosis two, both these chromosomes will move into this cell, and this cell will receive no, and whereas this is a kind of normal meiosis two. What will happen? Extra chromosome missing, normal, normal. So this is the process of non-disjunction. This happened during the meiosis one or meiosis two, during the anaphase or telophase. Due to this case, you can see over there can be a loss of chromosome or a gain of chromosome than the actual natural numbers. The next one is the point mutation. Mutation altering the sequence of DNA nucleotide. If involve only one or few base pairs alterations in coding sequence, then called the point mutations. So there could be the point mutations in this regard. So for example, some point mutations occur due to the spontaneous pairing and pairing errors that occur during the DNA replication. So if a few bases are involved, the alteration of few bases are involved, then this is called the point mutation. Other form, the damage to the DNA caused by the mutagens. Mutagens are the factors, the chemical, the substances, or the radiation responsible for the process of mutation. This could be the environmental factor, the external factors. So the changes or the mutation could be internal or external. So for example, this is the diagrammatic representation. This is, for example, a chromosome, or this is showing the basis sequence A, B, C. So it means that this is the normal condition: A, B, C, gene A, gene B, gene C. So normal B protein is produced. For example, the protein is B protein. Point mutation, the base substitution, for example, there is addition or there is substitution of another base over there. So the B protein will be inactive. If there is the insertion of another, you can say there is a kind of another insertion of another base, or other, you can say the gene, the B protein will remain inactive. Or if there is the deletion of this part or removal of this part, the B protein will be inactive. But for example, there are the gene changes in gene position A, C, B. B gene or proteins regulated because of the change in the gene position. For example, that protein was manufactured or prepared by the gene B. It's okay. Gene B is present, but another location that protein will be formed. But if there is a chromosomal rearrangement, for example, this will become another chromosome, this will shift towards the another chromosome, the B gene or B protein will be inactive. So what is the summary? Base deletions, additions cause the frame shifts in the code. What are the frame shifts? Their effects are so large that protein becomes useless. For example, if the base are deleted or edited in the sequence, they will cause the frame shifts, and it means that the effects are so large that protein become useless or inactive. Because sometimes deletions or addition may create a stop or triplet codon or code part way through the gene, so the protein will never made at all. If there is a stop codon for, then there will be the stop signal for the messaging of that protein, and there will be the stoppage of further protein making. If there are the base substitutions, they have no effect at all. They will cause the silent mutation. Because many amino acids have more than one triplet code, and if one base is changed, one amino acid is still coded for it in certain cases. There are two kinds of mutations: frame shifts and the silent mutations. Example of the mutation is the sickle cell anemia. The normal, you can say the hemoglobin is actually providing the oval shape to the or biconcave shape to the RBCs because hemoglobin is a globular protein. It has four polypeptides, 2 alpha and 2 beta. Actually, in the normal condition, there is the mutation in the 1 beta chain in case of the sickle cell anemia. In normal hemoglobin, normal person, the beta chain consists of this sequence: valine, histine, leucine, threonine, proline, glutamic, lysine. But when the person had the sickle cell anemia due to the mutation in the beta chain, this sequence goes wrong. One base has been changed, CTT into CAT. What will happen? Instead of glutamic acid, that is normal condition, another amino valine will be there. So this will cause the sickle cell anemia. In sickle cell anemia, due to distortion of a beta chain, a beta polypeptide, the whole structure of the hemoglobin will disturb, and the normal RBCs which have the oval or biconcave shape, they will turn into the sickle shape, and this will cause disturbance in their transport. So there will be no supply of oxygen to certain areas of the body. The next example is the albinism. Albinism is the autosomal recessive. So it means trait. It means that it will convey itself only in the or represent itself only in the autosomal recessive homozygous condition. One type of albinism is different. That is, what is the? It is affecting the eye, not affecting the skin. That is sex linked. Now, what is actually happens in the albinism? Albinism is very rare. What happens actually? There is the loss of pigment of that is melanin pigment in eyes, in hair, in skin. So their eyes appear to red, they have lost the skin color, and they have lost the hair pigmentation. So this is the normal these are the normal symptoms of the albinisms. So there are two types of albinism. One is the autosomal, other one is the sex linked. The sex linked is very rare. What happened in this case? There is a gene, tyrosinase. Its effect is actually that it converts the dopa into um, the dopaquinone into the melanin, so melanin will become active. So if the gene is not properly is for example, it is absent or inactive in some cases, in case of the albinism shown by the autosomal homozygous recessive condition, so there will be no gene and no conversion into the melanin. What will happen? So that's why the melanin will not be produced. So in case of the plants, tyrosinase is also present. The example is the blackening of the slice of potato or the apple when it is exposed to air. The next one is the Huntington disease, that is due to dominant allele. And this is the neurological disorder. Dominant allele means that if a person is heterozygous, then that person will have that disease. Uh, in this case, there are involuntary movements and the progressive mental deterioration in that person. It will have the loss of brain cells and the ventricles. These are the spaces in the brain will become so large. The symptoms appear in the middle age, and what happens? Mutation in the chromosome 4 that is responsible for coding for the Huntington protein. If the person has no Huntington disease, what will happen? The segment that is present in the chromosome 4 that is responsible for the coding of the Huntington protein, it consists of small repeats of CAG triplet, CAG, CAG, CAG. This is the normal condition. But if the person is diseased, that typical segment on chromosome number 4 will have the large number of repeats of CAG, CAG, and CAG. More these repeats of CAG are called the stutter. If more stutters are present, earlier the symptom conditions will appear. More stutters mean more CAG repeaters are present, then more disease condition will be shown earlier. So this is all about the Huntington disease.